Okay, class, what we're going to talk about is reproductive physiology and anatomy. Basically, what happens is, in an overview fashion, is basically this. The dad contributes sperm. The mom contributes the egg. The dad in the sperm will have 23 chromosomes after forming the gamete. The gamete is formed from a process called meiosis. The egg the same way, formed from a process called meiosis, reduction division, which basically means in the gamete you went from the individual that had 46 chromosomes to 23 in the egg and 23 in the sperm. Not any 23, but there would be one from pair 1, one from pair 2 chromosome. We'll talk about that. Okay. If fertilization occurs, the first cell to be formed is called the zygote. That particular cell is a totipotential stem cell. It can form anything. Once it is formed, if fertilization occurs, then a rapid sequence of mitosis will occur because remember, the human has trillions of cells. The first 8 weeks in the mother's womb, in utero, is the embryonic period in the human, first 8 weeks. After that, from the 8th week until the time of parturition, whenever that is, then it is a fetus, and after a fetus, once it's born, it's a neonate, and the neonatal period is the first 28 days. So that's kind of an overview. So basically, genes from 2 individuals are combined in a random way to produce a new individual. That allows for genetic variation and adaptability to a changing environment. Okay? It is through this process that we get adaptation to our environment. Okay? Germ cells, also called germination cells, sperm and the egg, which are in the gonads, the ovary and the testes, come together and produce the 46 chromosomes, original number, and then we're back to where we mentioned. Okay? So right here you can see the adult male in the testes produces sperm, the adult female ovary produces an egg, an ovum. If fertilization occurs, you get the zygote. Then the zygote will do a bunch of mitosis to get a bunch of cells to make either female or male. If it's a female, it would have been an XX. If it's a male, it's an XY. Autosomes and sex chromosomes. Okay. What happens is we have something we call a karyotype. And basically what a karyotype is where you obtain the chromosomes from an individual, and you lay it out in a certain pattern that has been internationally determined on this pattern. Okay? So we have different chromosomes, each has different genes on it, kind of like a file cabinet. We're having different files for several file cabinets. So the way a chromosome is determined visually primarily is by how long it is, the length of the chromosome, where the centromere is, which holds the 2 pieces of genetic material together, and what is the staining pattern. The first 22 on this called a karyotype. A karyotype, the old term for nucleus used to be karyon. So what a karyotype is, to type the nucleus. So on a karyotype, where you've laid them out and taken a photograph of them, the first 22, internationally determined, this is determined by cytology, how you want to lay it out, are called the autosomes, and the 23rd pair is for the sex chromosomes. In this case, this is 2 X's, which would be a female. So each zygote gets 23 chromosomes from mom and 23 from dad. produces 23 pairs of homologous chromosomes. Now, what homologous means the same type. So that means, let's say this one came from dad, this one came from mom, but it's a one. So you have the same genes. Heterologous means opposite. So if I put a 1 with a 7, that's heterozygous, homologous. Okay, but this is homologous chromosomes, homologous. The other be heterologous, and so that's what we're looking at. The 22 pairs are autosome, the first, and the 23rd are called the sex chromosomes. Each zygote has 2 alleles of each gene because one allele came from mom and one came from dad. Okay? An allele, by definition, is an alternate form of a gene. So mom would give an eye-color gene and dad would give an eye-color gene, to say it another way. Now, in most cases, both alleles can be expressed. Then we'd have to get into recessive and dominant determinations and everything. But in most cases, both will be expressed. However, an allele can be silenced by epigenetic changes. Now, what an epigenetic change is, so again, as I was alluding to, each zygote has 2 alleles of each gene. But in most cases, both are expressed. But an allele can be silenced by something called an epigenetic change. Now, epigenetics, you can look that up, is outside the genetics, above the genetics. And what it is, you aren't actually changing your genetic code. You're actually silencing some genes by chemical action. The chemical action would be to put something that blocked the expression of the gene. And so that could be something like methyl groups or zinc fingers. And so when the gene when it's read, attempts to be read, it can't when you're trying to make the messenger RNA, shall we say. So what will happen though is, if a gene is silenced, okay, mom had eye color, dad had eye color, if a gene is silenced, then you can't express that gene. So therefore, it would be only the other parent's gene that could be expressed. So if it was mom had blue and dad had green, and green was blocked, then only blue could be expressed, even though eye color involves a whole lot more than just one gene, to be very honest. I'm just using it as an example. Now, however, when fertilization occurs, though, you remove that silence. So in the zygote, all the chemicals would be removed, so everything could be expressed, except in a case of what we call genomic imprinting. In genomic imprinting is where it's a blocker that is not removed, so it won't be even expressed in the offspring. Because with just regular silence, it could not be expressed in the individual itself, but could in the offspring, because you would have removed that in the zygote. So that's what we call genomic imprinting. The sex chromosomes, females have 2 X's, okay, and a male is an X and a Y. So in other words, when fertilization occurs, the female the only possibility she has is to pass one of the X's. Now realize, she got one of her X's, though, the mom, the female, from dad and one from her mom. But she could pass on an X. The male has an X and a Y. So all that male can pass on would be an X or a Y. So essentially, if he passes on a Y, then it is a male. So really the determination of the sex is based upon the sperm, the contribution of the sperm. Now, the X and Y chromosomes look different. They look different. Go to that picture. Here's the X, here's the Y. The Y, actually, we know evolutionarily, was derived from the X. So the X has about 1,090 genes, while the Y only has 80. The Y chromosome the main thing about the Y is it has genes to determine maleness. Genes to determine maleness. So we all kind of develop along, to say it tongue in cheek, the female line until the Y chromosome kicks in those genes to turn on hormones to make us become males. Okay? The testes specific genes, those are the ones that are imparted, that must kick in to make us male, the males, are located in a palindrome area. Now, palindrome in words means it reads the same way in both directions, like madam, N-A-D-A-M, N-A-D-A-M. So on the palindrome, they read in the same direction either way you go, which assists in repair. I won't get into that. That's more of a general biology, to assist in repair in problems that can occur. Okay? Here again is a picture of the X and the Y. Okay. In females, one of the two X chromosomes is inactive. Okay, let's look at this. Now, let's go to this picture. Okay, let's say that's two X's. Okay. Let's say that's two X's. Now, the X chromosome, which is considerably larger, has a bunch of genes on it that are imparted for things in life, like development of some of the clotting factors and so forth. The Y chromosome could not have anything really, per se, should we say, imparted in the development of life itself, because then, think about it, the female would be at a disadvantage because she wouldn't have those genes. Okay. However, on the other hand, a female has two X's. Now, if there are genes in there imparted for life, like the gene to make certain clotting factors, then what could happen is, if there was a mutation in one gene, in one X, the gene to do this, more than likely the other X, which came from the other parent, would be okay. So the person would be fine. The female would be fine because she would have a second copy. The male would not, because he would only have an X and a Y. So a bad X then would be shown in the male primarily, like hemophilia, to give an example. But to equal the playing field, this normally is what happens. One of the X chromosomes in the female becomes inactive. And what happens is that inactive one actually is inactivated and sticks to the corner of the nucleus. See, this is the cell, but that's the nucleus. And that corner sticking becomes a condensed chromatin, what we'll call a heterochromatin, is termed the bar body. The bar body. So she actually, in each cell, would be working off one X. Now, here's where what we call a mosaic, like a shirt that has multiple colors is a mosaic, is that in some cells, the bar body would be from the dad's chromosome, and in some cells from the mom. So that lady would be working from either one, but it would depend on which one. So it's more of a mosaic pattern in the male. So not all the genes in the bar body are inactive. Not all of them. Some are not. Okay. Now, if you may recall that in neutrophils the genetic material is called up. So when it's called up and you call the nucleus up, sometimes that inactive one can be at the tip here, known as a bar body. That means I know it was from a female because here's the And the term for this is called a drumstick.